Good afternoon. My name is Michaela Williamson, and today I will be presenting the paper that I chose, which was titled Phosphoglucose Isomerase Deficiency in Escherichia coli K12 is associated with increased spontaneous mutation rate. Listed here are the authors, and to give a brief overview of the paper, they focused on an E. coli strain with a depleted PGI gene, meaning the PGI gene was removed from the E. coli. And they wanted to see how this affected the mutation rate. Was it increased, decreased, and what else was affected along the way? So to start with some background information, I wanted to look at what phosphoglucose isomerase is, which is also referred to as PGI. PGI is a key enzyme in early glycolysis, which is the breakdown of glucose. PGI catalyzes glucose 6-phosphate, which is also known as G6PH, to fructose 6-phosphate, which I have a picture shown here to the right showing that. The coli K12 strain used had a depleted PGI strain, so that PGI strain was removed. Under normal physiological conditions, E. coli chromosomal DNA accumulates amidori products, which are referred to as APs. APs are just more stable ketoamines. Those APs then are converted to advanced glycylation end products, which are referred to as AGEs. And here in my figure, I have the glycylation process, which shows that you start with a protein and sugar, also known as Schiff bases, and then due to structural changes or conformational change, those are when your amidori products are going to be produced. Then those, like I said, the AP products are converted to the AGEs. DNA modification with these AP products and AGEs leads to spontaneous mutagenesis in E. coli, which is just a change in the genetic information. The main question that they wanted to look at was, does the E. coli strand containing the depleted PGI gene accumulate higher amounts of G6-phosphate? Does it grow slower? And does it exhibit higher mutation frequency? And in order to look at this question and go through the methods and try to answer these and get the results, first you need to make sure that the PGI gene is removed from that E. coli strain. So to me, that was the most important part, and that's what I chose to focus on for my presentation. The first method used was PCR, or polymerase chain reaction. They wanted to isolate the E. coli BW28357 chromosomal DNA and also isolate the PGI depleted strain. So they wanted to use the one that they removed the PGI gene. They wanted to isolate that. Here I have listed the important items that are needed for a PCR reaction to be done correctly. So I thought I would just put those up there as well. And then also the steps for PCR, which are denaturing, annealing, and elongation. So for denaturing, you want to make sure the DNA is put to higher temperatures, around 95 degrees Celsius. For annealing, those temperatures are going to be put down lower to around 55. And then for elongation, you're going to raise the temperature just a little bit to in the 70s, but not quite as high as the denaturing step to complete the PCR reaction. From there, once you have your PCR samples, you can use an agarose gel electrophoresis to view your results. So here I have a figure just showing, here's the agarose gel and the electrophoresis tank. Your wells that you're loading your PCR samples into are near the negative electrode, which is hooked to the negative in the power supply, and then the positive is hooked to the positive electrode here. Once you turn the power supply on, those samples are going to run towards the positive side, allowing you to view your results from that PCR reaction. The next method that was used was Western blot. Once you've completed your gel electrophoresis, you then transfer that to PVDF membranes. From there, you block nonspecific sites and use primary and secondary antibodies to target your protein and antigen that you want to see. After doing that and from there, you are able to detect and analyze your results. Like shown in this figure, you analyze your final Western blot results, and it shows all the steps prior, starting with your sample preparation from the PCR. So here are the results from the PCR. It is indicated that the PGI gene was deleted in the E. coli strain of BW28357. They used a kanamycin resistance known as CAN-R in the paper, referred to as CAN-R, which is just shorter than the displaced chromosomal DNA that they removed from the PGI gene. So here in this figure, under 1 is your parental, so the PGI gene is still in the E. coli strain. The paper said that there was about 1,891 base pairs in this sample. And here next to the DNA ladder that was used, which is in the middle under M, that's 2,000. So it is very close to the parental, showing that the PGI gene is still there. For 2, which is on the right side here, that is your strain where the PGI was deleted. So they refer to this as the knockout, and that was about 1,568 base pairs. So that was shorter than the one with the PGI gene, which did indicate that the PGI gene was deleted. And this thus confirmed the PGI gene deletion. Next to the Western blot results, it was important to know they used an antibody against PGI, which shares 59% homology with the E. coli enzyme. They did get some positive results here shown. Under 1 is the parental, like in the last figure, so there was PGI present, so they did expect to see a positive staining result. Under 3, they used yeast PGI, which was with an anti-PGI antibody. So that was used as a positive control, so they did expect to see that result. Under 2 was the negative staining result because there was no PGI gene. So there was no reaction with the anti-PGI antibody, so thus there was no positive staining result shown. What does this mean? It means that the PGI gene was successfully deleted from that E. coli strain, so then they were able to further use methods and find results that led them to know that the accumulation of G6-phosphate was shown in the PGI gene deletion of E. coli, along with delayed growth and increased spontaneous mutation. The DNA of E. coli PGI deficient strain, so no PGI was present, they were less modified with carbonyl derivatives. They did not expect to see this result. They actually expected it to be more modified, so that was really interesting to see that they weren't always correct with their hypothesis. And then the protein extract from the PGI proficient, so where the PGI gene was still in the E. coli strain, it did catalyze the release of G6-phosphate, which they did hypothesize. So the last thing was the involvement of PGI and the repair of G6-phosphate derived lesions in DNA. They know that they do play an involvement with each other, but they aren't sure what exactly that is. So that leads us to what's next. What are the future things they want to look at? Why PGI travels to the nucleus is currently unknown. Based on their research and findings, they might have come up with one idea of why it does localize in the nucleus, but they want to look more into that and do more research before they actually say this is a reason. And then the involvement of PGI and repair G6-phosphate damaged DNA, they know that there is an involvement with the PGI, but they don't know exactly what it is once again. So they need more research to be done, and they want to look into that more for some future research to be done. And like I said, I chose to focus on the PCR and the Western blotting because without that PGI gene being deleted, they could not have moved further with their investigation or their research to get these results. And here is my reference. Thank you.